Hi everyone, it's Dr. LeBlanc with another video capture lecture on personalized medicine. Our learning goals are to understand how knowledge of a patient's genetic makeup can lead to better medical treatment, to understand what a microarray is, to understand how knowledge of the genetic changes that lead to an individual patient's cancer can direct specific treatment, to understand what pharmacogenetics is, to learn about personalized medicine divisions of hospitals, and to understand how genetically based tests can lead to faster, more precise targeting of medicine for infectious diseases. So, is there a one size fit all drug therapy? Or would it be better if we looked at our drug therapies and the patient's genetic makeup so that we could target specific drug or treatments for an individual patients? Personalized medicine is the use of information about a patient's genotype and other clinical data in order to select medication or therapy that is specifically suited to that patient. Personalized medicine has become a very important aspect of healthcare. One area in which personalized medicine has become very important is in molecular profiling of tumors. Tumors are classified by appearance under the microscope. This may or may not reveal underlying genetic changes, and sometimes it might lead to misdiagnosis and incorrect treatment. Molecular profiling involves identifying the genes causing the development of the cancer. Microarrays can be used to identify the genes in a tumor whose expression correlates with each other, and it may lead to treatments targeting those genes. DNA microarrays can be used for molecular profiling. Researchers can compare cancer cells to normal cells, and they can also compare two different types of tumors. They can identify groups of genes that are turned on in the cancer cells and off in normal cells, and other groups of genes that are turned off in the cancer cells and on in normal cells. What is a microarray? Well, a microarray is a 2D array of DNA sequences, actual DNA molecules, from thousands of different genes. Each spot on a microarray has many copies of the same gene. You can allow messenger RNA or cDNA from a sample to hybridize to a microarray, and it will only hybridize to sequences where it is complementary. You can measure the number of hybridizations per spot as well as which spots get hybridized. So you can get information about which genes are turned on or turned off and how much they are turned on or off. This is an actual microarray. You can see this is on a microscope slide. This microarray contains thousands and thousands of different genes that can be analyzed all at the same time. Each spot on a microarray contains multiple strands of an identical DNA molecule. The DNA sequences in each spot are unique. So this spot has different sequences than this spot, for example. Each spot represents a single gene, or even one exon of a single gene. Thousands of spots are arrayed in orderly rows and columns on a solid surface using a robot. The precise location and the sequence of each spot is recorded in a computer database. Microarrays can be the size of a microscope slide, like shown here, or even smaller. Once a microarray experiment is completed, a computer-generated graphic can be analyzed. In this graphic shown below here, the red squares represent highly expressed genes, and the green squares represent genes that are not expressed. This microarray is comparing two different tumors. two different origins of diffuse large B-cell lymphomas. Okay, so the arrays are the same, but the messenger RNAs that were hybridized to do the arrays are different. Here we have the genes that were expressed in a germinal center B-like tumor, and here the messenger RNAs from an activated B-like tumor. So if we look through this array, we can pick out a few genes that we might want to pay particular attention to. Here, for example, is a myosin 1C right along this row here, and we can see that it's expressed in the germinal center cells, but not in the activated B-like samples. Okay, it's off in those. If we look a little at some more of the spots on the array, we can see these genes seem to be turned off. All of these genes seem to be downregulated in the germinal center B-like samples and upregulated in the activated B-like samples. So we can see that we're seeing very distinct differences between these two tumors that look the same but have a different origin. How important is this information? Well, if we look at the survival rate of patients with a B-like germinal center B-like cells or activated B-like cells, we can see that the patients who have the germinal center type have a much higher survival rate. So if we can target some therapies to these different groups of patients, we might be able to improve their prognosis. Pharmacogenetics is the study of genetic variation that causes differing responses to drugs. This is a big issue in the drug industry. If you remember that first graphic I showed you where you're giving a pill to a whole bunch of different people, well, all of those different people can have different responses to the same drug. And it's based on things like absorption, distribution, variability in the target molecule. Patient genotype is important information for finding the correct drug dosage. Here's an example. Thiopurines are a class of immunosuppressive drug. TMPT is an enzyme in cells that inactivates the thiopurines. Genetic variants of TMPT result in reduced activity. So if a patient has normal TMPT activity, it's going to inactivate at a slow but regular pace the thiopurines. And that's okay. You just give more drug and you pace the dosing. However, if a patient has an inactive TMPT, if you gave the same amount of drug as you're giving to a normal patient, the thiopurines will build up inside the cell and have an overdosing effect, too great an effect. So this information is very important for proper dosing, efficacy, and effectiveness of the drug. Proper drug dosage depends on a variety of factors, including the following 5 points. The rate of transport of the drug from the digestive tract into the metabolism, into the bloodstream. Okay, so here is our digestive tract. How quickly it goes from the digestive tract across the epithelia of the digestive tract and into the bloodstream is very important. The rate of transport into the cells where the drug actually acts. Here, it's the blood vessels are going in between the cells. How quickly does it get from the blood cell into the actual cell where the drug needs to act? The ability of the drug to affect the function of the target protein. The ability of the drug to be metabolized by the liver. And finally, the rate of excretion of the drug from a body are all very important considerations. These 5 factors are affected by genetics because the proteins encoded by the genes are directly involved. For example, transport proteins for uptake of the drugs into the bloodstream, right here, for uptake of the cells of the drug into liver cells, hepatocytes right here, for uptake into specific cell types, crossing the blood-brain barrier, getting into kidney. All of these proteins on the surfaces of cells are important in this transport, and they all can have genetic variants that will affect the process of transporting the drug across the membrane, either into or out of the cell. Many major teaching hospitals and other hospitals now have personalized medicine centers that are an integral part of providing top-notch healthcare experience for the patients who go there. This is the Partners healthcare system, which is a healthcare system in Boston. It's linked to Harvard Medical School and other medical teaching hospitals in that same area. If we go to their website, you can see that they have a lot going on in their personalized medicine division. There's the Laboratory for Molecular Medicine, tests, ordering, reporting, resources. There's translational genomics core services, frequently asked questions, contacting translational genomics core, the biobank, which is a very important resource for many physicians in the area. If we go down here, there's a lot of very interesting videos and links that you can find out a lot more information. The biobank is very, very interesting resource. The biobank has available for researchers high quality, affordable human samples that other researchers can use. It provides researchers access to high quality consented samples to help foster research and advance the understanding and causes of human disease. There's a lot of career opportunities and genetics training programs associated with Partners Healthcare. So it's definitely a resource to try and check out. What about infectious diseases? Infectious diseases are cause a lot of healthcare problems. They're very prevalent in hospitals and in other contexts. The tests for different infectious diseases were often slow. You had to take samples from a patient, you had to culture them, and you had to perform a lot of different tests that could take a long time. Recently, panels of PCR probes have been developed in order to detect specific infectious diseases. For example, Clostridium difficile, or C. diff, is a very prevalent infection that people who are on antibiotics for a very long time can often acquire. There's a very rapid PCR method which can detect the toxins that the C. diff puts out to tell a healthcare provider very quickly, in approximately 2 hours, whether or not a patient actually has this particular bacteria. There's been a bit of debate in the literature about the use of these, but by far and away, the consensus is that these multiplex panels are testing not just C. diff, but a variety of different pathogens are overall terrific because of their cost, their effectiveness, and their speed. Now, what about nutrition? One of the hot topics in the fall of 2015 was the DNA diet. Is it for real? or is it a fad? Well, you can find a lot of popular literature on them, and you can also find a lot of websites touting the benefits of DNA diet. This is an article from Men's Health, for example, where they tried the DNA diet, and here's another website, many, many websites. The question is, of course, are these real or are these fad? Well, you're educated enough in genetics now that you should be able to go to these websites and make that determination on your own. In summary, personalized medicine uses information about a patient's genetic makeup to target drug therapy. Microarrays can be used to highlight molecular differences between similar tumors or cancers. This knowledge can inform the healthcare professionals as to which treatments would be best. Pharmacogenetics examines an individual patient's differences in drug uptake, transport, and tissue-specific metabolism. Personalized medicine divisions at major hospitals are responsible for analyzing a patient's genetic makeup and prescribing individualized treatment. PCR-based tests can diagnose common infectious diseases accurately within hours. And is the DNA diet authentic? You be the judge.